Hello and good evening everyone. So are you guys ready to listen to our presentation on lipoprotein? Let's start it. So, what is lipoprotein? Lipoprotein is spherical macromolecular complex of lipids and specific proteins called lipoprotein. So, what is the function of lipoprotein? First, it keeps water-insoluble lipids in aqueous plasma, and it provides mechanism for transporting lipid components to and from the tissue. Do you guys know what are the class of lipoproteins? There are chylomicron, low density lipoprotein (LDL), very low density lipoprotein (VLDL), high density lipoprotein (HDL), and free fatty acids.
Now, let's move on to the role of lipoprotein lipase in the lipoprotein catabolism. Before that, let us introduce ourselves. I'm Rose.
And I'm Shamira.
Hi! What is the first role of lipoprotein lipase? Lipoprotein lipase is used to hydrolyze triglyceride. The triglyceride in chylomicron and in very low density lipoprotein, or known as VLDL, are hydrolyzed by this enzyme. This enzyme is found in the capillary wall. So, the final product of this catabolism are fatty acids and glycerol.
The next role of this lipoprotein lipase is to promote the cellular uptake. This enzyme will promote the cellular uptake of chylomicron remnants, cholesterol-rich lipoprotein, and also free fatty acids. This process requires ApoC2 as a cofactor that enhances the saturable uptake. Then, nearby cells can pick up the free fatty acids and glycerol molecules to use for energy.
So, there are two roles of lipoprotein lipase, which are to hydrolyze triglycerides in chylomicron and VLDL, and also it will promote the cellular uptake. So, remember!
Now we will explain further about the lipoprotein catabolism. Lipoprotein metabolism can be divided into two pathways, which are the exogenous and the endogenous pathways. So, what is meant by exogenous pathway? The exogenous pathway refers to the absorption and transport of dietary fat and cholesterol. Absorption of fat and cholesterol in the intestine is facilitated by bile acids, which are synthesized in the liver. Recall that bile acids are synthesized from cholesterol, represents a catabolism pathway for cholesterol. However, normally greater than 95% of the secreted bile acids are recycled back to the liver through the enterohepatic circulation, and so little cholesterol is lost through this process.
The dietary fatty acids and cholesterol are transported into intestinal enterocytes. They are converted into triglycerides and cholesterol esters. Then they are packaged into chylomicrons and released into bloodstream. In the bloodstream, chylomicrons bind to the lipoprotein lipase (LPL), which is a lipolytic enzyme expressed on the endothelial surface of capillaries in muscle and also fat tissue. LPL will be further discussed later on. Upon binding to LPL, triglycerides in the chylomicron are hydrolyzed to produce free fatty acid FFAs and glycerol. The FFAs are then taken up into muscle for ATP biogenesis or adipose tissue for storage. As chylomicrons become depleted of triglyceride and relatively enriched in cholesterol, they are then transformed into chylomicron remnants and taken up by the liver. Take note that remnants are then used in the formation of HDL, which stands for high density lipoprotein.
Endogenous pathway. In endogenous pathway, liver synthesize and secrete very low-density lipoprotein, also known as VLDL, which is containing cholesterol ester, and triglyceride, which assemble from fatty acid derived from adipose tissue or synthesized de novo. Similar to chylomicrons, VLDL particles will bind to lipoprotein lipase to produce free fatty acids. Meanwhile, approximately half of the LDL remnants will undergo endocytosis directly by liver, while the remainder of VLDL will convert to lower density lipoprotein or LDL via hepatic lipase. Here's some information for you. First, LDL receptor is the only receptor capable of clearing significant amounts of LDL from the plasma. Second, half-life of LDL in the circulation is only 204 days. Third, the majority of LDL receptors are expressed on the surface of hepatocytes, and so the liver is the primary responsible for the removal of LDL particles from the circulation.
Now, let's talk about the activation of the lipoprotein lipase. What is the substance that activates the lipoprotein lipase? Lipoprotein lipase is activated by the glucose-dependent insulin. Glucose-dependent insulin is also called as the gastric inhibitory polypeptide because it secretes in the intestine. How does it occur? It's occurred because the glucose-dependent insulin can regulate the cell survival. It deposited the 3-3-L1 leukocyte. In the presence of insulin, lipoprotein lipase and the triglyceride accumulation ability increase. So that is how the insulin helps to activate the lipoprotein lipase.
Lipoprotein lipase inhibited by insulin under roles of lipoprotein lipase. Insulin will increase glucose uptake by the muscle. The effect of insulin on adipocytes is to stimulate them to produce and secrete lipoprotein lipase. But the effect of insulin on skeletal muscle is to inhibit the secretion of lipoprotein lipase. This is because one of insulin's vital roles in the body relates to fat storage. It inhibits the breakdown of fat cells and stimulates the creation of body fat. Insulin tells the body to stop burning its fat stores and instead absorb some of the fatty acids and glucose in the blood and turn them into more body fat.
Okay guys, now it's the end of the video and let us recap with me, Aileen and Safia. Okay, now, do you guys remember about lipoprotein metabolism? As we all know, lipoprotein metabolism can be divided into two pathways, which are endogenous pathway and exogenous pathway. Do you guys remember what is the exogenous pathway? Exogenous pathway is the absorption and the transport of dietary fat and cholesterol. While endogenous pathway is the breaking of VLDL in the liver from triglycerides and cholesterol acids. Okay, now, do you guys remember what is the role of lipoprotein lipase in lipid metabolism? There are two main roles of lipoprotein lipase, which are to hydrolyze triglycerides, chylomicron and VLDL, and also to promote cellular uptake. Lipoprotein lipase will be activated by glucose-dependent insulinotropic, and it is inhibited by insulin by increasing the uptake of glucose by the muscle. Yay! Well done, guys! Hope that you guys guys understand about the role of lipoprotein lipase in lipoprotein metabolism?
I know what you're thinking. We'll be talking about the members who took part in doing this slideshow. Here's a quick overview of who does what. For the introduction, Siti Nurhaliza and Siti Nur Amira. For Azlin Hambali and Chen Zixin, they have done the overview of metabolism of lipoprotein, exogenous pathway. Shuhada has done endogenous pathway. Role of lipoprotein has done by Shamira and Rosma. Abdullah has done how LPL activated. And not to forget, Nasrullah has done how LPL inhibited. And for conclusion, Safia Amira and We did it!
We did it!
We did it!
Oh yeah!
Don't you love biochemistry?